Welcome back to this educational channel called Learn Bio with Janet. In the previous video, we discussed the process of mitosis. So let's go on to discuss cytokinesis in animal and plant cells. Cytokinesis is the division of the cytoplasm. So this subtopic is subtopic 6.2, and this is the third video in a series of 4 videos. So this is part 3, and the syllabus is the KSSM Form 4 syllabus, chapter 6 on cell division. There will be a hot question at the end of the discussion, and model answers will be given. So let's begin. We have already discussed mitosis. What is the difference between mitosis in animal cells and plant cells? Now, during mitosis in animal cells, centrioles are present, which help to form the spindle fibers that pull sister chromatids apart during anaphase. So these black lines represent the spindle fibers, and this structure here are the organelles called the centrioles. All right? So centrioles are cylindrical shaped organelles or rod shaped organelles that contain the microtubules or small tube-like structures. Their function is to form the spindle fibers, okay, represented by the black lines here, that are attached to the centromeres of the sister chromatids. So at first the sister chromatids are attached together in metaphase. Then during anaphase, the centromere will divide, and the sister chromatids will be pulled to the opposite poles of the cells. They will separate and move to opposite poles of the cell when the spindle fibers contract. Okay? So in this way, there is equal distribution of the sister chromatids to the 2 poles and to both daughter cells, to make sure that the daughter cells are genetically identical to each other and also to the parent cell. So the centrioles are very important because they help to form the spindle fibers in animal cells. All right? And spindle fibers are important to ensure that the sister chromatids separate and move to opposite poles. Now, plant cells, however, do not contain centrioles. So how do they carry out mitosis? Well, they are still able to form the spindle fibers during mitosis. Although how they do it, we do not have to know. Okay? They do not contain centrioles, but they can form spindle fibers. How about cytokinesis? Cytokinesis is the division of the cytoplasm that happens after the nucleus is formed at the end of telophase of mitosis. So cytokinesis occurs after mitosis, or it may overlap a bit with the telophase of mitosis. So when telophase is going on, cytokinesis may already begin to occur. Okay? Now, cytokinesis is different for animal and plant cells too. Just like mitosis is different for animal cells and plant cells. So what are the differences? Let us discuss cytokinesis in animal and plant cells to find out. Firstly, let's discuss cytokinesis in an animal cell. How does it occur? So imagine a cell that has just undergone telophase of mitosis. Okay, so 2 new nuclei are already formed. Now the cell has to divide into 2 to form 2 daughter cells. Okay, so there is division of cytoplasm. So at first it's just a cell, oval-shaped cell, and then the plasma membrane constricts in the middle of the cell between the 2 nuclei. Or we say the cell constrict. Constrict means to become narrower. So the cell will become narrower in the center here, between the 2 nuclei. So how does it constrict? Now in the cell there are many microfilaments which can contract. So microfilaments are thin filaments or tube-like structures that contain a protein called actin, okay, that can contract. All right, so you contract, the microfilaments will contract in the center of the cell. We call it a contractile ring of microfilaments. So as the microfilaments contract, a cleavage furrow is formed. Cleavage furrow is like a groove, that goes inwards. It will become deeper and deeper inwards as the microfilaments contract more and more. Okay, so this is a keyword. There's formation of cleavage furrow in cytokinesis of animal cells when the microfilaments contract. All right. So microfilaments at point of constriction contract to form a cleavage furrow. So in this way, the cell constricts. Constrict means to become narrower. Becomes narrower here, narrow and narrow until in the end it splits to form two daughter cells. Okay? So that is how cytokinesis occurs in animal cells. Microfilaments contract, form cleavage furrow, cell constricts and splits into two daughter cells. Now, how about cytokinesis in plant cells? It's very different because plant cells are separated by cell walls. All right, two plant cells will be separated by a cell wall. So a cell wall must form in between the two nuclei to form two new cells. Now, how to form the cell wall? So at first there are vesicles. Vesicles are small tiny bags that contain certain substances inside them. So vesicles will gather in the middle of the cell between the two nuclei, in the center of the cell between the two nuclei during cytokinesis, and then they will combine to form a cell plate. Now this is the cell plate formed when the vesicles combine. Then the cell plates are surrounded by a new plasma membrane, okay, right here. And a new cell wall substance is also formed among the spaces of the cell plates, like the spaces here. So what this cell wall substance is, we don't have to know. Now, the cell plates expand outwards. Okay, so they will expand downwards here and upwards here, until they combine with the plasma membrane. So this red line here represents the plasma membrane, okay, of the original cell. Here they have combined the plasma membrane, and lastly, cellulose fibers, okay, are produced by the cells to strengthen the new cell wall here. So the cell plate has developed or become a new cell wall, okay, and in that way, two diploid daughter cells are formed. Okay, a summary. Vesicles combine to form the cell plates. Okay, vesicles are like bags that contain substances inside, small bags or sacs that contain the substances inside. And then vesicles combine to form cell plates surrounded by a new plasma membrane, and then cell plate extend or expand outwards until they combine with the plasma membrane. Cellulose fibers are produced to strengthen the new cell walls here. Two diploid daughter cells are formed. Let us discuss this important question, which may come up for structured questions as a structured question or an essay question. Question 1. Compare mitosis and cytokinesis, or just compare cell division in animal cells and plant cells. Okay, mitosis and cytokinesis, or just simply cell division. Okay, so we may be asked to compare these two processes in animal cells and plant cells. 6 marks or 5 marks. Now let's look at the similarities. So if you are asked to give a comparison, you must have both similarities and differences. Each similarity is 1 mark, each difference is 1 mark. So have a combination of both that adds up to 6 marks in this case. Okay, similarities. in both animal and plant cells, spindle fibers form. Whether they have centrioles or not, spindle fibers can form, which are used to pull the sister chromatids apart so that the sister chromatids will move to opposite poles in nuclear division, okay, or in mitosis. Secondly, in mitosis, there are 4 phases: prophase, metaphase, anaphase, and telophase. Now here I didn't have enough space, so I just put PMAT, but in the exam, you have to spell out the 4 phases. So for both animal and plant cells, mitosis consists of the 4 phases. Now, 2 daughter cells are formed from one parent cell in mitosis, or in cell division in both animal and plant cells. Now let's look at the differences. In an animal cell during mitosis, centrioles, as shown here, are present, which are used to form the spindle fibers. Okay? Plant cells do not contain centrioles, but they can still form the spindle fibers. Right? Okay, now animal cells. During cytokinesis, microfilaments like these brown fibers here, or filaments here, will form and they will contract. As they contract, they will pull the plasma membrane inwards to form the cleavage furrow, which is the depression shown here. Okay, the point that moves inwards, goes inwards. So that is the cleavage furrow. It's a keyword. Microfilaments and cleavage furrow. But in plant cells, there are no microfilaments, so no cleavage furrow is formed. Okay. Instead, for plant cells, because there should be a cell wall. Plant cells have cell walls that separate the cells. Thus, to form the cell walls, first vesicles will gather at the center part between the 2 nuclei, and the vesicles will combine together to form the cell plate, which then expands outwards, okay, downwards here and upwards here. Then it will develop into a cell wall that separates these 2 parts to form 2 daughter cells. So in animal cells, the vesicles, cell plate, and cell walls are absent. Okay. Now, if they ask for comparison of mitosis and cytokinesis in animals and plants, not just the cells, but in the body of the animals and body of the plants, then we can say that cell division occurs in tissues throughout the body of animals, but cell division occurs mainly at the meristems or shoot tip and root tip of plants. This is for the location where cell division occurs. Where does it occur in animals and plants? Okay, so this may be relevant or not relevant to the question here. Depends on how it is asked. If they ask for difference between cell division or mitosis and cytokinesis in animals and plants, then you can give this as part of the answer. But for the rest, it's important for you to memorize all these. I think there are 6 points here. 3 similarities, 3 differences. Or maybe if you can think up of more, you can write it down. Okay, so the top 3 similarities and top 3 differences are important in answering this question. So before we go on further, I would just like to make a clarification of the previous video where we talked about telophase. In mitosis, the process, video number 2, I circled number 1 as one of the important events. In telophase, the chromatids arrive at the opposite poles. And I also circled number 5, nucleoli and new nuclear membranes are formed. Now we can also actually circle number 3 and number 4. Chromosomes uncoil and form fine chromatin threads, spindle fibers disappear. These 2 events are also important in telophase. Alright, so there are 4 points in telophase that can be stated if they ask what occurs in telophase. Okay, but if they ask you for the behavior of chromosomes in telophase, then it is number 1, chromatids arrive at the opposite poles for the behavior of chromosomes. And number 3, chromosomes uncoil and form fine chromatin threads. These two are the behavior of the chromosomes. Okay, kelakuan chromosome, things that happen to the chromosomes in telophase. Okay, the other one, number 4 and 5, concerns the spindle fibers, nucleoli, and nuclear membranes, which can also be used to explain the events that occur in telophase of mitosis. So circle up number 3 and 4 too. Now let's discuss mitosis in the onion root tip. How does it look like? So this is an experiment that can be done in class, where the root tip of an onion can be treated and stained with some kind of stain, so that the nucleus will be stained, and after that the root is squashed, squashed flat on the slide, and if you observe under the microscope, you can see the cells of the root tip in various stages of mitosis. Alright, because the root tip is a type of meristem, where the cells are actively dividing by mitosis to produce new cells for growth. Okay, so the root tip contains cells that are undergoing mitosis, about 1 mm of the root tip. Okay, that's at the tip of the root. That's the place where mitosis is occurring. So let's look at some pictures or some observations under the microscope. All right. For example, you may see the cell looking like that, where you can see that the chromatin, is becoming thicker, and you can observe that there are thread-like structures, but not so vague, but it's becoming clearer and thicker. So this may be early prophase. And then if you see a structure like this, where there is a center line, this shows that the chromosomes are lined up at the metaphase plate, or we call it the equatorial plate. For KSSM syllabus is equatorial plate. For KBSM syllabus is called metaphase plate. Both are accepted. Alright, but we follow the textbook. Okay, so this shows you the metaphase plate, or the equatorial plane here. So this is the stage of metaphase, where the chromosomes become aligned at the equatorial plane. Now, this observation shows you that the cell is undergoing anaphase, where the chromosomes are moving towards the opposite poles, where the centromere moves first, and then the two arms of the sister chromatid will follow. So this is anaphase. It's very clear-cut here. And these two may be already reaching telophase, and cell division may have occurred already. So 2 new cells are formed. That's all for this video. In the next lesson, we will continue with the subtopic 6.2 on mitosis, and it will be the 4th video in the series. In this next video, we'll be discussing some questions, objective and structured questions on mitosis. Model answers will be given too. This video is very important for you to go through and to try, as it will help to reinforce your learning, so that you can master this topic well, this challenging topic of chapter 6, and it will help to improve your exam grades. So thanks for watching, and stay tuned for the next video. Please share, like, and subscribe. Goodbye.